This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiommunity, recorded on February 7th, 2018. I'm Kevin Bonham, and I'm joined, as we have for the last couple of times, by Camilla Engblom.
Hello.
Hello, and making her return performance back from a long winter of doing nothing important that I can tell.
Oh, the
Kate
long
Franz.
winter of our discontent. Yay! I'm back. You can't get rid of me.
Welcome back.
Yeah, because we tried so hard to get rid of you by constantly sending you texts and emails.
You tried so hard to get rid of me by scheduling them over my thesis writing, my defense, my move to California, my 3 moves throughout Boston in the past year, my job hunt, and starting a new job. So,
Sounds like a breeze.
Yeah, I definitely don't think Kate deserves any of the blame for this. We have no idea where Matt is at the moment, but Chadine was going to be joining, and then apparently like everyone in her house just started vomiting all at the same time. So she's not going to be here. And we'll miss her. But I think the three of us, we're going to attempt between the three of us to have a cogent discussion. May or may not be possible, considering that only like 1.5 of us read the paper. But we're going to give it a shot.
Oh, I also wanted to add, I was writing a paper, putting a paper through revision and getting it published as well, on top of all that other stuff.
Oh, that doesn't sound important.
Yeah, that stuff too.
Well, okay, well, last week... Oh, I should also mention, this episode may be coming out before the last episode that was recorded. While I was out of the country, Matt, Camilla, and Chadine recorded an episode, and there was some technical difficulties that we're trying to sort out. But this episode might come out before that one, where they discussed Camilla's awesome cancer and bone paper.
This is just what happens when you're awake, Kevin. Everything falls apart.
I think that's true.
Kevin leaves behind some sort of virus, so
Yeah,
we're
I think that
completely and totally crippled without him.
crashed my audacity. Such audacity.
Yeah, I definitely have that power.
Yeah.
As a computational biologist, I control all things computer related.
Yeah, you could convince me.
Before we actually get into the paper, first up, we should say what everybody's drinking. Kate, since you're coming back from the dead, why don't you tell us what you're drinking tonight?
Oh, you're going to love this, Kevin. It's Diet 7-Up.
What?
So, well...
Plus gin? Plus vodka? What?
No. See, I'm 3 hours behind you because I live in California now. And...
Oh, yeah.
I'm just saying, like,
3:30.
And you can't drink at 3:30?
Not on a Wednesday.
All right, fine.
Technically, I'm at work.
I see.
Yeah.
Interesting.
Yeah. So I'm going to save the drinking for later tonight.
Understood. What will you be
fair.
drinking tonight then?
I will be drinking Line 39 Sauvignon Blanc. It's my new favorite $8 wine, which replaced my old favorite $8 wine, Canyon Road Sauvignon Blanc. Which... if you attend Harvard Immunology seminar, I often call pineapple wine. To the great confusion of the bartenders there, except for the one guy who's the bald bartender. That guy's my boy. We're best friends. He was very sad to see that I was no longer going to be at immunology seminar, but we said our goodbyes. He knew what it was about, and he also believes in Canyon Road Sauvignon Blanc. But I've replaced it.
Are you going to tell us what pineapple wine means? Because I have no idea.
Yeah.
It tastes like
neither.
pineapple.
Oh,
sorry. I thought that was like kind of obvious.
No.
I call it pineapple
been a long
wine
day.
because it has a fruity, crisp taste, much like a pineapple.
Fantastic. And Camilla, what are you drinking?
Well, I thought to honor the snowstorm that's happening in Boston right now, which is more like a snow mixed slush. Maybe, Kate, you missed this, I'm sure. I'm drinking some Sam Adams Summer Ale. Yeah, because my fridge is very updated.
Fantastic.
Yes.
This evening, I'm drinking a sort of... I didn't have time to stop at the liquor store on the way home, and I'm out of beer. So I went into the liquor cabinet and I got out some Short Path gin. Short Path is a distillery that's in Boston that opened up by the climbing gym that we go to. And so I'm having a Short Path gin, and my wife insisted that I not say gin and tonic because it's not tonic, it's just seltzer. But the gin itself is really tasty. And so just getting a little carbonation is totally enough. It's really tasty.
That sounds great.
Sounds perfect.
I love gin.
I'm just going to cut out your awkward responses.
It's way more sophisticated than my summer ale.
Mm-hmm.
Grown-up points to Kevin, for sure.
I also wanted to mention our Patreon subscribers that are donating $21 now per episode for us to make this podcast, which is phenomenal. It means that we can not only pay for web hosting, we can buy
a round of PBR at Franz.
We can buy a round of PBR at Franz. Franz is the lovely little dive bar next to Harvard Medical School. But it also allows me to pay for the audio editing software that I'm using. And as we discussed on the last podcast when it was just me and Matt, to encourage Matt to drink despite the fact that he has a small child at home, we're hoping to maybe pay for a beer for at least one, if not more, of our hosts every episode. So your donations are going to a really, really good cause when you support us at patreon.com/Audiommunity.
Seems like a great idea. We have 2 parents with small children as hosts, and we should encourage them to drink as much as possible.
I think so. They're both responsibly in 2-parent households.
That's right.
And so one of them can take over while the other one, gets a little buzzed at the end of a Wednesday. I think that's perfectly reasonable.
That's fair. This is their hour of science and not parenting.
Yes. In terms of rewards, to be perfectly frank, though, we actually would love to be providing something extra for our Patreon subscribers because it's really awesome that you're supporting us. I mean, it encourages us to record more often, but everybody gets those rewards. So yeah, if you have ideas, send them along. If you have papers that you want us to discuss and you're a donor, we are almost certainly to discuss it.
Yeah. If you have a presentation you'd like me to give you a critique on, we can set up a little Skype chat. I'll just tear it on down. It'll be great.
I don't know that you want to offer that, but that's a very generous offer, Kate.
I love doing that.
Kate will also make you a figure in PowerPoint if you really ask her nicely.
Okay, I have excellent PowerPoint skills, and
I know you do.
I will not have you talk about my PowerPoint skills with such derision. Okay.
What derision, Kate?
Okay,
here's the deal. That was totally sincere.
Here's the deal. Look, I'm not one to plug myself like this, but go into PubMed, type Kate M. Franz, and go look at my molecular cell review. There is a beautiful picture of cells undergoing or deciding between 2 different fate decisions that I drew fully in PowerPoint. It is gorgeous. It was the front page, the front banner page of the molecular cell website for the 2 weeks that it was posted. It was wonderful.
I don't know if this will translate into audio recording, but I just want to.
Oh, is that a clap?
It's a very slow clap. It's a beautiful story, though, Kate. Beautiful story.
Yes, lovely.
It was wonderful. I was so proud. I took a screenshot of the molecular cell website just as my own personal validation that the skills are real.
It wasn't your personal validation. You sent it to me immediately and bragged about it.
Can I just question how are you using PowerPoint and not Keynote? Is that just me?
I don't like Keynote. It's just weird. I grew up with PowerPoint. I've adapted to it.
The real question is why you haven't learned Illustrator, despite the fact that you have several people around you that would be happy to help you learn.
Illustrator costs money.
A wonderful set of YouTube tutorials by yours truly that could help you make wonderful Anyway.
We are sponsored by Apple and Microsoft and
the real tragedy is that those videos have gotten a couple thousand views, and I am not collecting the 25 cents that I would have had I monetized. But oh well.
Oh well.
You can watch them without ads.
Yeah. I also don't need to make figures anymore. So there is also that.
What are you doing with your life now? I think our listeners want to know.
Oh, okay. Yeah. So as I mentioned at the top of the show, I recently graduated, finished my PhD, got my paper out, moved to California, got a job. I pulled off that little hat trick there, getting everything all together at the end. So currently what I'm doing is I'm working for a science advocacy organization called the Organ Preservation Alliance. And what we do, you can think of it like American Cancer Society, except we're much smaller and more targeted in our focus, which is to preserve organs outside of the body. And so, you can imagine the applications are obviously to organ transplant. If you could preserve organs for longer, we believe that you could do more transplants because you would no longer be limited logistically. But additionally, if you could preserve cells, tissues, and organs outside the body, we think that you would have a lot of benefits to developing technologies and developing therapies. So we're trying to work towards this goal of preserving organs, and so that's what I've been doing. So I'm a program director. Yeah.
I just want to say that when you started talking about that, your whole demeanor just changes when you go into professional Kate mode. I'm part of an organization that, and it was really nice, but also it just totally incongruous with the rest of your commentary.
I don't think that there's that big of a difference between professional Kate and shooting the crap with people Kate went to grad school with.
There's a pretty big difference, but we'll let our listeners decide. This evening, we're going to talk about a paper that I definitely read in great detail, and it's called "Strains of Bacterial Species Induce a Greatly Varied Acute Adaptive Immune Response: the Contribution of the Accessory Genome." And the first author is Yuri Sella, and the last author is Vincent Fischetti. And this is published in PLOS Pathogens. When was it published? July of last year, of 2017. And so this paper is sort of addressing a question that is somewhat obvious in some ways, and also a little bit understudied. And that is the fact that different people respond to pathogens in different ways. So that's sort of intuitive. You think when I get a cold, it's not necessarily the same severity as when my wife gets probably the same cold. And most people traditionally have thought of this in terms of interindividual differences in immune response. So we've talked about MHC on this program a lot before. This is one of the main ways that our adaptive immune system responds to pathogens. And the MHC is a very diverse gene throughout the human population. There are a lot of different versions of MHC that are presenting different peptides to T cells, both cytotoxic CD8 T cells, but also to CD4 helper cells, which will change the B cell response. And one of the ways that you could potentially explain why different people respond to pathogens in different ways is the contribution of their personal immune system, MHC in particular, but also other factors. What this paper is looking at instead is the contribution of the diversity in the bacteria or the pathogen, which in this case is a bacterium. And so the idea here is that though we often talk about I got an influenza infection or I got a Staph aureus infection, not every flu is created equal. Not every staph is created equal. There's a lot of variation in the type of strain that you're infected with, and this might have implications for the immune response. You guys are being very quiet, and it's a little bit troubling.
Sorry. Because I have a real job, I haven't read the paper, so I was just thinking about what you were saying.
You were explaining it so well.
Oh, great.
No, I think
I turned into a listener instead of a host. Sorry.
That's right. So would you like us to come with some questions, Kevin?
No. I think Matt tends to interrupt me more because he has points that he wants to make. And so I'm just a little bit troubled by the fact that I'm speaking uninterrupted for more than 60 seconds.
Got it.
You're welcome.
And I do the same to him. I don't want to malign Matt too viciously here because I do the exact same thing to him all the time.
Well, I think you covered the main point, right? I mean, it's really the paper is set out to investigate what happens, like what if we take the cells from the same person, do those cells respond differently to different subspecies of different bacteria, rather than saying, okay, do different individuals respond differently to the same bacteria?
Right, exactly. And I think it's worth defining. So in the title, they say the contribution of the accessory genome. And I think it's worth sort of talking about what we mean by the core genome versus the accessory genome.
Yes, please.
So for organisms like humans and mice, for mammals and non-prokaryotes, basically, in general, different individuals of the same species are going to have basically the same complement of genes. Obviously, there's variation within populations. So for an individual gene, we mentioned MHC already, there's a lot of variation in that particular gene, but everybody's got several different MHC loci. And they have basically the same number of MHC loci. Everybody has a hemoglobin gene. There's probably less variation in that. There might be some in the population, but everybody has that gene. And that's true for the vast majority of genes in our genome. Even if we have different versions of that gene, we all have the same set of genes. With bacteria and other pathogens like viruses, that's not necessarily the case. And in particular, for bacteria, different strains, even of the exact same species of bacteria, they can have up to 30% of their genome can be completely different. In other words, you have like 30%, one strain of an individual bacterium might have 30% of their genome that consists of genes that are not present in other strains of that exact same species.
So how do you really define a species then? If it's so much variability within a species, where do you draw the line of one species versus another?
That's a great question. And it's actually pretty complicated and somewhat controversial. So for animals, for humans, for bears, for mice, the way that we define species is often, it's a little bit functional. In other words, one way that you can define species is to say that organisms that are capable of reproducing and producing viable offspring constitute the same species. Mm-hmm. So if you have a bunch of different dogs, for example, you have lots of different types of domesticated dog, but if you breed them together, they can produce viable offspring. And when we say viable, we mean an offspring that itself is capable of reproducing, as opposed to there's examples of species that can mate and produce offspring, but those offspring are often sterile. So a great example is
ligers.
Ligers, yeah, lion and tigers. I was thinking, I don't actually know if ligers are sterile, but certainly if you mate a horse to a donkey, you get a mule, and mules are viable, but they're not. They can't actually reproduce with either horses or with donkeys. So we think of horses and donkeys as separate species. For bacteria, it's a little bit more complicated because bacteria don't reproduce sexually in general. They produce by binary fission. And so you don't have this idea of the ability to produce fertile offspring. And you have this problem of giant accessory genomes, sometimes giant, sometimes there are minor differences, but often you have wide variation. You also have this problem of horizontal gene transfer, which I think I've mentioned before, but this is basically like microbes can share chunks of their genomes in accessory pieces like plasmids that are separate from the main genome, but they can also share chunks of DNA that get integrated into their main chromosome. And this can happen across very, very large phylogenetic differences. So bacteria that are from different phyla can actually share genes. It doesn't happen very often, but it can happen. And so how do you actually define a species? One of the ways that you can do it is you can say, for a given species, let's look at just the genes that actually are fundamental to that species. In other words, if we look at 1,000 different strains of E. coli, all of those E. coli, even if they have a lot of variation, there's going to be some set of genes that is common across all of E. coli. And we can look at those genes and we can say how similar or dissimilar are they from one another.
So is the idea here that you're trying to measure time? So like you're trying to measure the evolutionary time between the different species of bacteria?
If you're looking at the core genome, if you're doing what I'm describing, then yes, that's the idea, is that the more similar so one of the genes that is often used, not looking at the whole core genome, but looking at one specific gene that a lot of studies have used to define species is something called the 16S ribosomal RNA gene. So this is a gene that codes for the RNA portion of the small subunit of the ribosome. The ribosome is the machinery that produces proteins inside cells. And the reason that this gene is useful for for sort of checking the relationship of bacteria is that every bacteria needs to encode a 16S ribosomal RNA gene. Most bacteria, not all, but most only have a single copy of that gene. One of the things about having a flexible genome is that a lot of times genes can get duplicated. And if you're talking about multiple copies of the same gene, it can be tough to infer phylogeny from that. So generally speaking, the 16S rRNA gene only has one copy. And because it's not coding for a protein, it evolves much more slowly than most protein coding genes because for something that encodes for a functional RNA, there are no what we call synonymous mutations. So for proteins, because of the way the codon because of the way that codon translation works, some mutations don't actually produce a functional difference in the protein that they encode for. But for the 16S ribosomal RNA gene, all of the base pairs of the RNA have some function in terms of how they shape that the product. And so there are no synonymous mutations, and so it tends to evolve very, very slowly.
So the 16S RNA then would be part of the core, for sure, part of the core genome that you were talking about before.
Right. And it is also present in bacteria of all types, basically across the entirety of prokaryotic space. There are 16S ribosomal RNA genes. So we can sort of use this one gene as a proxy for the relatedness of all bacteria. But for within an individual species of bacteria, there are a lot more genes that are going to be common across that entire species. And we can use that to define, we can use the similarity across all of those genes to get a sort of a higher resolution picture of the relatedness of bacteria. It can be complicated a bit by the fact that you have horizontal gene transfer, and horizontal gene transfer happens more often between closely related bacteria. So that can cause problems, but in general, that's the idea.
I mean, it kind of makes me think of trying to parse out whether a cell is one single cell or a doublet in single cell RNA-seq when you investigate the transcriptome of the different cells. You know, if two cells have stuck together and you're trying to figure out if it's just one or if it's actually the contribution of two, because in this case, if you have a bacteria, but you have horizontal gene transfer from another species, it might look like the other species, but you're actually looking at another strain of bacteria. I guess it's a lot more complicated than it looks on paper.
Yeah, that's definitely true. And that can be a huge problem when we're doing like metagenomic sequencing. It's a problem that the lab that I'm in now deals with a lot. In the case of the bug we're going to talk about in this paper, Staph aureus, it's not as big a deal because we have a lot of isolates sequenced. So what that means is that we've isolated basically single cells by plating them out And then we grow those up, and then we can sequence individual colonies, and those represent a single lineage. And a lot of these have been sequenced, so we have some sense of the sort of sequence space occupied by strains of Staph aureus. And by aligning those genomes to each other, we can get a pretty decent picture of the evolutionary relatedness of different strains of staph. But it's worth noting, and it's important for this paper, that different strains of Staph aureus, even though they're the same species, some strains may have genes that other strains do not. And that's going to matter as we walk through this paper.
As a non-bacteria person, can you just, before we delve into the paper, talk about what a prophage is?
Oh, sure. So one of the bacteria, like us, have viruses that infect them. Viruses that infect bacteria are called phage. And many, if not most, phage are integrative, meaning when they infect a bacterium, they can actually integrate their genome into the genome of the bacteria. And many phage have different phases of their life cycle. One of them is called I always mix them up, but there's the lysogenic, and then there's the what's the other one? Lysogeny, and it's confusing because lysogenic doesn't actually lyse the bacteria. I think it's confusing.
Lysogenic and lytic?
Lytic, yes, thank you. And so the phage can actually integrate into the bacterial genome, not replicate, not produce new virions, and then as the bacteria replicates, the phage genome gets replicated along with it. When the phage is integrated into the bacterial genome, that is called a prophage. So it's when the phage is integrated into the bacterial genome, but isn't actually generating new virions. And when it switches into the lytic phase, this means that it's going to basically replicate and then bust the bacteria open to spew new phage into the environment.
Cool. So it's just hiking along with the bacteria, basically, and letting itself be replicated and contributing to that bacteria, but not making more of itself separately.
Yeah, exactly. This is analogous.
Yeah, I was going to say, it's analogous to retroviral or lentiviral integrations into mammalian genomes. So they also are replicated through the DNA synthesis machinery. So like when the daughter cell, when the cell divides, you get 2 copies of the virus as well. And then in the bacteria, different stimuli can activate that phage. It can exit from the bacterial genome and begin its replication cycle to lyse the bacteria and release new phage.
Got it.
It's really, so some like extra bonus homework looking into the cell fate decision of the lambda phage lysogeny lytic. That decision is really, really interesting science. Something that got me super interested into viruses as a young child.
How young? As a 2-year-old, I started thinking about.
I believe Kate was doing science at 2 years old,
like 10th grade.
Awesome.
The Campbell's biology chapter on microbial genetics. Awesome. Very interesting to me as a kid.
I feel like I had that experience with a cancer book as well. It's like, all of these things can go wrong when the cell divides. How do we even stay alive?
Totally.
That's awesome.
All right. So basically, figure 1 of this paper. Actually, before I do that, I should say, I thought that we should do this paper. Chadine recommended it, and then I read the abstract, and I was like, oh yeah, we should definitely do this paper. And then I read it, and I was a little bit disappointed, if I'm being perfectly honest.
Okay.
But when we get to figure 1,
this, and I have an idea of what this paper is about, and it's not based in any sort of knowledge of reading it, just looking at what the experiments were and guessing. Mm-hmm. So I have many questions to test my guesses.
Okay. So figure 1, they sort of lay out the paradigm that they're going to use for the rest of the paper. So basically, they have a bunch of different strains of Staphylococcus aureus, which is an opportunistic pathogen. It lives on the skin of a lot of people, but it can also cause really severe infections. You may have heard of things like MRSA, which is methicillin-resistant Staphylococcus aureus, or multi-drug-resistant Staph aureus. There's also various flavors of really, really awful Staphylococcus bacteria that are resistant to a lot of the potential therapies that we might use to treat infections.
Yeah.
It's sort of like
your hands.
Yeah, right. It's sort of in the media as like flesh-eating bacteria is often Staph aureus. But Staph aureus is a pathogen that causes a lot of different infections, but as I said, it can also hang out on your skin and not cause infection. And so there's quite a wide variety of responses that we might have to this bug. And so one of the things that this paper is sort of trying to do is to say, are there differences in individual strains of Staph aureus that might explain why certain strains will launch into these really, really severe infections?
Yes. Question 1.
Yes.
Why are we looking at T cells and B cells and not dendritic cells?
I don't know. I'm guessing because these are adaptive immune cell biologists, not innate. Still have that question.
I think it will
definitely be relevant for the experimental design. But basically, in Figure 1, what they're doing is they're taking blood cells from a donor. So they take peripheral blood, meaning they can basically draw it from the arm. And then in that peripheral blood, you have what are called mononuclear cells. So this includes T cells, B cells, monocytes, and neutrophils, and a bunch of other things like that. And then they basically incubate these cells in vitro with a bunch of different strains of Staph aureus. And then they quantify by facts whether the T cells and B cells proliferated, whether they started expanding. And proliferation for cells like T cells and B cells is a measure of antigen-specific response. So when you are seeing cells that go into this rapid division stage, that means that those cells had an antigen-specific response to something in the Staph aureus.
Yeah, and I think it's a really important thing to mention. You meant when you were talking about the different types of cells that are included in these peripheral blood cells, so the PBMCs for short. And probably there are not so many neutrophils in there. I think they get separated out, but the monocytes are there. And the way that the assay is set up is really that they give the bacteria to the cells in vitro, and then they wait for 4 days, and then they give the bacteria again. So presumably there can be some presentation of the antigen. The monocytes can actually present antigen to the T cells in this in vitro culture. And so
these are bulk PBMCs,
yes,
that are getting stimulated. Okay.
Yes.
Right.
And they also give an anti-CD8 antibody to these cells. So you're basically triggering T cell activation by giving the CD8 antibody. CD28 antibody, sorry.
Yeah, I should say, thank you for the clarification. Neutrophils are not mononuclear. That's one of the reasons they're not included, because they actually have like these weird bipartite nuclei. Exactly. I forgot about that. All right. So yeah, so that's an important feature to think about, that they have plenty of time to be stimulated. So even naive cells that are present in the blood, because they have this sort of 4-day window and then they hit them again, even naive cells that were in circulation could be stimulated by this assay. And what they find basically is that different strains, they test, 10 or 12 different strains of Staph aureus, and there's a pretty wide variation in how much of an immune response is generated by these different strains.
Second question. Do you think that there is a wide variation? So you look at if...
Yeah.
So it's just from 0 to 20% proliferation? Is that significant?
I think that's quite a lot. If you have none of your T cells proliferating versus a third of them proliferating, I would say 30% of your T cells proliferating in response to something is quite a lot.
Okay.
Yeah, I agree with that. Yeah, I think it's pretty remarkable variation.
Yeah, I have no metric for this.
Just think about it this way. Like, would you expect 30% of your adaptive immune response to have antigen-specific responses to a single pathogen?
No.
Right.
Okay.
And I guess that when they're proliferating, they also become 30% of the population because there's...
Yes.
Mm-hmm. Yeah.
Yes, certainly. Certainly that's true because they're blasting and being responsive. But yeah.
That never stops being funny to me.
What?
The blasting?
Blasting. It's ridiculous. Anyway,
it doesn't even strike me as odd.
That's a callback.
Just rolling around in my petri dish blasting.
All right. So then they give like some representative facts plots of what they're looking at. I feel like that could have gone in the supplement, but whatever. The next thing that they do is they show us. So that was the first figure is just PBMCs from a single donor. But then what they do is they look at 10 different donors. They do the same assay with all of these different things, and they sort of look at the average response. And what they see is a trend where if their first donor responded to a strain, there's a fairly consistent response with other donors. It's not like a perfect correlation, but it's fairly suggestive. The mean response across the 10 donors to the strains that the first donor responded to is pretty high.
Yeah, I think it's pretty nice separation. You have the same pattern across the different strains.
Yeah, the main objection I have to this is like, why didn't they just start with the 10 donors? Like including the response to the single donor as if they did that first and we're like, oh, there's a difference. And then we did more like that.
Well,
that doesn't strike me as super useful.
What I like with the single donor is that you're really asking, with the same cells, do we have a different response? Because in figure 1C, you have the 10 different donors, but you don't see how each patient tracks for every group, right? So you don't see if the same kind of trend is there. But if you look at the single patient or your actually healthy donor, you just have to infer
it from the difference in dynamics.
Yeah.
That, okay, one of these patients, like, because they didn't respond very well to RN 4220, but there is a response for the next strain, one of the non-responders started responding, but you don't really track it. So.
But there are ways of tracking that for individuals. So you could have the plot in 1C where you're looking at,
but now it's just like data. Like some people just have PIs who are very specific in the way they would like you to make a 20-panel figure.
Do you and I have any experience with that sort of PI?
I don't know. I don't know any examples, but sometimes that's a thing that people really want.
Speaking of figures, I have to say, if I was a PI on this paper, I know I'm weird in this case, but like you really, really should not just export a figure from Excel that includes like gradients on your bar plot because the color gradient, it doesn't actually do anything for the figure. And there's like drop shadows on the legend. Did that bug anyone else? It really bugs me.
This is no Kate on PowerPoint.
Now that I'm looking at it and you mention it, I won't be able to not see it.
The worst is the drop shadow on the like significance bars that come later on.
No.
Let's see where in like figure
4.
Figure 4. Yeah, exactly. There's drop shadows on the little bars that say like whether it's P less than 0.5.
Not to get
just
too
bugs the shit out of me.
Because I recently went through the proofs of my paper and like saw all the inconsistencies that I never noticed. But I see this now very brightly in figure 4.
Yes, the not significant.
The not significant bar is thicker than the significant bar.
Yep.
And I was just like, oh no. Yep. I just imagine that the first author didn't see that. And then when this paper went out to publish, they're like, why did I put that in 1 point when everything else is in 3? What was I thinking?
It's awful. It's awful. Anyway, so one of
the can I ask a question here? Because one of the things I was thinking about these different strains of bacteria here is how do these numbers mean anything to you in terms of like, aha, 8325, yes, that's a really common one, or USA 200 is more pathogenic, or, you know.
No, but I'm not a Staph aureus biologist. It might make it. These might be very clear to other people, but no, I have no idea what these are.
Yeah, because it was
the breakdown that they give us is that like which ones are MRSA, which ones are like vancomycin resistant, but beyond that, no, I have no idea what these mean.
Yeah, because I'm really curious to know whether some of these are more likely to be found on your skin, or some of these are more likely to be pathogenic, because that would be a nice correlation with the data that they have, right? If one is more prevalent than the other, maybe that can actually relate to how much of an immune response you have a priori.
Right. One of the questions I had at the end of this paper is like, could this difference be explained by the fact that some of these strains are just more prevalent?
Yes.
And so if people have been exposed to these strains more, then you would expect them to have more circulating antigen-specific responses to those particular strains, and that wouldn't be necessarily super interesting. And I think they go some ways towards addressing this later on. But let's just jump down to figure 2. Basically, they're showing, again, using the one donor as an example and then checking it in the other 10 donors, what types of cells are proliferating. They show differences in Th1 versus Th17 responses. I feel like this is probably interesting to like Staph aureus biologists, because if your immune response is skewed towards Th1 or Th17, that can have very strong differences in terms of outcomes. But I was thinking I would just sort of breeze past that figure for time, and because I think it's not super critical to the overall point of the paper. Does anyone disagree with me on that?
No, I agree.
Okay. So jumping then to figure 3, here they're trying to get at mechanism. So this is where the sort of second part of the title comes in, the idea of the accessory genome. And one of the things that they're looking at in particular is those prophage. So those parts of the genome where a phage has integrated. And one of the things that distinguishes different strains of Staph aureus are the presence of different sorts of prophage. And so in particular, they're testing the Newman strain, and there's a couple of different serotypes of assay that they're using, one of which is M1. And for both of these strains, basically they generate versions of these strains where all of the prophage in the genome have been knocked out. They've removed the prophage from these strains. So they're identical except for the presence of these genes from bacteria viruses. And what they see is if they take the knockouts of these prophages, the response goes away. The proliferation of cells goes away, and the number of cells actually responding in this PBMC assay drops to basically nothing.
Yeah, I thought it was interesting that the T cell response went away, but not the B cell response. I don't know if you were thinking about that or if you reacted to that, because the T cell phenotype is really striking. Like you say, it just goes away completely. Mm-hmm. And then in C, you kind of see that the B cells proliferate the same and the IgG is made the same. And that was curious and interesting to me. I don't know what it means, but I thought it was interesting.
Yeah. So one thought is that because T cells can respond to a sort of wide variety of peptides, and they might be, I was sort of thinking about it in terms of like exposure. So you would expect that there are a lot of peptides that are going to be similar to the ones that Staph aureus has that are in commensals or not in the context of infection. And so those peptides might be sort of T cells specific for those peptides might get sort of cleared out.
Yeah.
Prior to encounter with this staph, although that doesn't necessarily explain why the B cell responses would not be affected.
I was wondering if it was an antigen presentation problem rather than on the T and B cell end. You know, that if the peptide presentation wasn't working properly when you knocked out these somehow, or but the whole protein was able to be presented. But I thought it was a little bit, I'm not sure actually.
So for those of us who don't think about B cells, what is the difference between T cell activation and B cell activation in this assay? So how does each one become active?
That's a great question. So I am not 1,000% positive in this context. From a sort of like basic immunology standpoint, typically the way we think about T cell activation, in particular CD4 positive T cell activation, is that you have an antigen presenting cell. In this case, it's probably a monocyte, picking up, phagocytosing some component of the bug, getting an innate immune signal through a pattern recognition receptor, and then presenting a peptide on MHC class 2 that some CD4 positive T cell is specific for. So that CD4 positive T cell is going to receive both signal 1 from the TCR MHC peptide interaction and signal 2 from the fact that the antigen presenting cell has been activated and is presenting co-stimulatory molecules. For the B cell, typically the way that we think about that in a sort of normal immune response in a lymph node or something is that it would have some CD4 positive T cell that's been activated, and it would basically present MHC on MHC class 2. Some CD4 positive T cell would recognize MHC class 2 on the B cell and then provide stimulation to that B cell. Mm-hmm. In this assay, I'm wondering if maybe the B cells can be triggered. I know that there are ways that B cells can respond in T cell independent ways through like TLR ligands. So in particular, like TLR9, I know is super strongly stimulating to B cells. Mm-hmm. So in this assay, I don't know if they actually require T cell help.
Mm-hmm. That would make a lot of sense to me based on
and maybe it's figures. Antigen independent for the B cells.
Yeah.
If they're getting like really strong TLR9 signaling from staph, and then they might just start proliferating in a sort of not necessarily antigen independent way, but like a lot of B cells have antibodies that recognize like polysaccharides and that are bacteria specific, and so you might get some confluence of that.
Yeah, and it looks like everything is just stimulated all together in vitro, right? So they get the bacteria, they get the CD28, and so whether the T cells in the dish that are being activated also activate the B cells, or if the B cells get activated directly,
right?
We just don't know.
So could you, given the model that you just set out, could you say that in this experiment, the phage in the bacteria that were deleted are either necessary for the activation, so like the activation of the immune cell, or are necessary to be presented to activate the T cell? So either like they're they're detected actual by antigen, like the PRRs, and they give like the activating signal, or they produce the peptides that are presented that are needed to activate the T cells.
I would say it's almost certainly the peptides, because these bugs are going to have a wealth of innate immune signals that are irrespective of the phage, right? So they're going to have LPS, they're going to have bacterial DNA, they're going to have a bunch of crap that the monocytes can respond to from an innate immune signaling standpoint. And actually, now that I think about it, the B cell response, it seems like it sort of has to be T independent because the T cells aren't getting activated. Yeah, it's not surprising to me to say that like the B cells wouldn't respond to the presence of this phage, because the phage DNA in the bugs probably isn't producing much that is going to be visible to the B cell, right? But the fact that they're responding, despite the fact that none of the T cells are responding, suggests that the B cell response is more or less T cell independent.
I would agree.
Sort of points to innate signaling to the B cell.
And what's a little bit interesting is that if you look at in figure 3 in C in the right panel, you have a really big spread among the IgG expressing cells. So it looks like the proliferation is really consistent between the two groups, whether you have these prophages or not. But the IgG expression, you see this big split between the ones that actually express IgG or not in the TB4 in the prophage mutant scenario. So that maybe is a little bit intriguing.
You're saying that the individual points are really spread out, even though the mean is not significant between the two groups. Yeah, that's a good point.
Maybe it means nothing, but maybe it is something. And maybe you see a little bit more diversity there in whether they're directly or indirectly activated from the T cells.
Yeah, that's an interesting point. Yeah, I have no idea. Could be.
But I think that also goes back to Kate's point in the beginning. Why didn't they look at macrophages or monocytes? Or, because maybe you wouldn't have seen any difference between any of these strains. And it's only because you have these differences in antigens and the T cell response that you actually would see these interspecies variability. But if you would look at monocytes and macrophages, I'd be really interested to know, actually, but maybe they would react exactly the same across the different species.
If you looked at like cytokine expression, or if you looked at other ways that the monocytes are going to respond, then you might get a better picture. ELISA is probably more expensive than FACS, and maybe this lab doesn't have experience with that. And also the cytokine picture in this sort of mixed cell assay is going to be really, really muddy. There's so many cells that are probably doing so many things. Yeah. So like in principle, you could like isolate out the monocytes from the PBMCs and then hit just those cells. But I don't know. That might be a little bit muddy. I think it's definitely a limitation of the paper, but it's not like it doesn't cripple it.
I think it's harder to look at, activation of myeloid cells by FACS. That's for sure. Right. Like to really say, this macrophage is now activated. Mm-hmm. Right. I am definitely of the mindset that it's really complicated and you need to look at different markers, different genes, different cytokines, and typically in isolation to really know. Like you don't have an interferon gamma in the same sense as for T cells. Right. Unfortunately.
Yeah, I mean, you have some cytokines that you can do with macrophages and monocytes through intracellular cytokine staining, but it's not the same. It definitely doesn't have the same level of clarity that you get from T cell activation and B cell activation markers.
They should do single cell RNA-seq, which I'm positive any author in this paper, if I were the reviewer and said that, would start hunting me down. But
yes, I'm sure that's true.
That would overcome a lot of these issues that we're talking about.
Yes.
This paper was also published middle of last year, and my impression of the single cell RNA-seq world is that it is just accelerating at a phenomenal pace. Yeah. And so like this paper, if it were submitted today, might very well plausibly ask for a single cell, but
I don't make that as a legitimate criticism. Like, they should have done X, Y, and Z. I only bring that up as a suggestion of if this were an issue, that this is another expensive way and time consuming way that you could start to get to our criticism.
Yes. Kevin, I don't know if you want to cut this part out, but I see that they published it on January 11th this year. I think it was submitted in July last year.
Oh, interesting. Okay.
For the sake of
we can leave that in.
Awesome.
That's a good clarification. Yeah. So it was submitted in July, but it was published on January 11th, 2018.
But yes,
it's nice that you get that transparency from PLOS. So that's kind of interesting. I think that figure 3 is really helpful to sort of alleviate potential concerns that this is just an epidemiological thing where it's like certain strains people have exposure to, and so they have bigger immune responses. That might also play a role. Like there's no reason that phage genes should be more immunogenic. It might just be that the phage is the thing that varies among strains. And so,
yes and no, it might be something about the way that they're exposed to the bacteria normally. Maybe they don't see the phage as often. So the context of it. So if you're exposed to the strain, maybe you see it in the context where you never see the virus. But now in this dish, the bacteria becomes stressed, the phage is released, and that is what drives the activation of the immune cells.
Yeah, I think that's totally plausible. There's no evidence in this paper one way or the other.
Yeah.
So it could definitely be that the phage is immunogenic, and that explains something about the differential response. It could be that the presence of the phage is just differentiating different strains, and people are exposed to different strains at different levels, so they have a different baseline of antigen positive or antigen specific T cells and B cells in the blood circulating for certain strains.
I also didn't. So I didn't read the paper, but
you've said that.
Do they talk about if there's a difference in survival of the bacteria in these two different? So
I think that bacteria is already killed before they add it.
Oh, is it heat kill? Oh, okay.
Yes.
Okay.
Yeah.
Okay.
Yeah. The assay is all ex vivo. Yeah.
Interesting.
And
okay, because I was going to say maybe there's a difference in the viability of the bacteria, and
stuff,
so they might under with the phage present, they might activate replication, and now in these bacterial infections with phage, maybe you have more bacterial killing and more activation, blah 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 blah. But okay.
No, I was also thinking about whether, like, the way that they kill them, how you standardize for exactly the same amount of bacteria that you add. Mm-hmm. Like, how challenging is that? Can you, if you say we added so and so many particles or bacteria to begin with?
They're just doing it based on OD, optical density. So when you grow bacteria, you can sort of look at the optical density of the media, and that's a proxy for how many bugs there are.
Yeah. Optical density is just a fancy way of saying cloudy.
Yeah, exactly right. It's at a particular wavelength. This is a standard thing. They do actually say in their methods, which I give them credit, because most people will just report like what they used and not say. They say that the OD was chosen because the bacteria are at their maximal proliferation stage and before secretion of virulence factors and superantigens. So I give credit for them actually saying that that's the case. There's some question in my mind about like the average proliferation versus individual cell proliferation, and that could vary even if like on average they're different. You might have individual cells that are like, with the phage, they're getting to a lytic stage. And so you have like, 10% of your bugs are spewing phage out. That might change things. I don't know. But I think it's not unreasonable the way that they did that. Yeah. It's pretty standard.
Okay, cool. And then like you said, the figure 3 is really the one that makes this paper cool, I think. The fact that they remove the phages and they see that there's difference in response goes away. And
it would have been nice if they had done the reverse experiment. So take one of their strains that does not cause a strong immune response and just like add the phage in and see if that is sufficient to generate a strong immune response. But again, like that experiment is harder than the straight knockout experiment. And I don't, necessarily fault them for that, but it would have been cool to see.
Yeah. And I was also thinking for the pre-exposure, they did one experiment where they took cells from mice and said, that they can see the same differential activation using mouse cells. But also there, you could say that maybe you had some pre-exposure to pathogens. If they really wanted to go die-hard experiment, like a germ-free mouse, and then take cells from those and then see if you would have...
Yeah, but there's problems with germ-free mice too. And like their immune systems are totally messed up. So.
That's a fact too, yeah.
And germ-free mice are freaking expensive as well. But anyway, so figure 3, I think, is sort of the crux. Figure 4, so before Chadine let us know that her house was covered in vomit, the one thing that she said is that she was very confused by figure 4. And to be perfectly frank, it's not a very informative figure. It's also like a really, really terrible offender of like export from Excel figure. So like I have trouble seeing past that. Okay. Like the gradients on the bars are bad enough. I already mentioned the drop shadow on the non-significant bars. That's just inexcusable. You cannot put drop shadows in a figure for a science paper. You just can't. But that aside, So what they're showing is what they're calling the coefficient of variation in T cell proliferation, and they're comparing it between different strains versus between different donors. So the question here is the variation in response explainable by variation in the donor that they gave the strain to, or is it explainable by variation in the strain of bug? So you sort of have like two dimensions of potential variation, and the question is which of those dimensions explains more of the difference in response. And to be frank, I think that the differences between A, B, C, and D are marginal. They say A, which is the difference in T cell proliferation, is statistically significant, while the variation in interferon gamma for Th1 cells, in B cell proliferation, and in IgG expression are not significant. They all have sort of the same trend. The error bars in some are just a little bit bigger. And I don't know. I think this is not a particularly informative figure and doesn't really tell us a lot extra. But
yeah, I would agree with that.
So, okay. So T cell proliferation strains. Okay, trying to like wrap my mind around what this actually is saying.
Yeah,
okay,
it's a sort of.
So the coefficient of variation is essentially just a measure for if we were to plot like all of these values like in a histogram, what is the standard deviation? So the larger the number, the more variable. The actual numbers were, or the values were. So like the strains are more value. Okay, so T cell proliferation, when you group them by strains, you have more variation in the actual numbers there. But when you group it by donor, there's less variation. So the donors will...
The donor response to the same strain.
The donor response to the same...
Okay, has less variation than the differences between strains from the same donor.
Okay, gotcha. Maybe that's why there was two different representations of the data. Okay,
right. And I think the reason. One of the reasons that this is problematic is because they have few. They have like 16 strains and they have 10 donors. So like, I don't know. Those numbers just seem very, very small to make any strong arguments. And particularly because the trend for every one of these is the same, but some are statistically significant and some aren't, suggests to me that really we just need a lot more strains and donors to make any cogent argument on this point.
But the trend does follow the oh boy, I just lost my word. The trend does
follow the conclusion that they're saying, which is that the strain variation.
Thank you, Kevin, for that save.
You're welcome.
Yeah. So we would hope that if you increase the actual numbers of strains and donors, that these might trend towards significance, as people like to say in talks, and that it's actually the difference in strains and not the difference in donors. So I don't know. I feel like this should have been a figure 2, or maybe a small panel of figure 1, just to go for your 20 sub panels.
Yeah, I definitely agree. I don't like. Or even like honestly, even a supplement. Like they have other stuff that suggests this information. This is like another way of measuring the same idea and sort of like putting a numerical statistical bent on it, which I'm a fan of. I just like it seems really weak for the conclusion to the paper. Yeah. Which like, that's it obviously made it in. Plus Pathogens is no slouch of a journal. And I think the paper is it's interesting. It's an interesting perspective on it. I really want to know, like, is this sort of idiosyncratic to Staph aureus? Is this true of lots of other bacterial infections? I mean, we know that variation in MHC can have huge impacts on inter-individual differences in response to particular strains for like viruses and stuff. So like, is this weird for this bug? Is this a common trend? Maybe this lab is working on that. I have no idea. But it's sort of a cool, suggestive paper. And, aside from my complaints about Excel exported spreadsheets or bar plots, it's really clear also that like different people did different plotting, because some of them are clearly done in Prism and some of them are clearly done in Excel. But
I feel like once you use Prism, you can never use Excel again to make a graph. True. I just walked towards Prism and never turned back.
I agree with that 100%. And these days, though, I do almost everything like programmatically. And I don't even use like, I've got just a giant CSV and then I'm using code to plot stuff. I sort of miss when I could use Prism, because Prism is really fun.
Computers now.
You miss copy pasting. Copy pasting.
I miss using Prism. I think using Prism is really fun. And I miss it. I just can't do it anymore because my data tables are like crash Prism.
Next time I do a flow experiment, I could send you the Excel spreadsheet and then the Prism, and then you can make nice little graphs for me if you want.
Oh, thanks. That's very nice of you.
Right?
I probably wouldn't. I would probably just load it into.
I feel like you have more important things to do also.
So that's the conclusion of the paper.
Yeah, I would say I buy the fact that when you have the same patient or healthy donor, the cells react differently when exposed to different. So T cells respond differently when you expose them to different strains of the same bacteria.
Yeah, and certain strains seem to be broadly immunogenic for lots of different subjects, which is. I don't think that they really nailed that down. Even with the knockout experiments, they're probably collecting the same donors from the same hospital or something. So like exposure could still explain that to some degree. Mm-hmm. But the fact that they were able to narrow it down to the prophage, as at least for the T cells, that's cool. And there's a lot of future directions for this work. And I think it's pretty neat.
Absolutely.
So unless anyone has anything else, this has been Audiommunity. And as always, we want to encourage you to reach out to us. There's a lot of different ways you can contact us. You can go to our website, immunity.org, EMMUNITY.org. There's a contact form there. You can find all of our old episodes. You can search on different topics. As I mentioned last time, I put a sort of tag cloud next to the posts. So you can, like, if you really want to learn everything about T cells, you can click on the T cell tag and find all of our episodes about that. If you should definitely go to patreon.com/audiomunity and kick us, $1, even 50 cents an episode would be really awesome. If you have ideas for what we should do with that money, you should also tell us. We're now at the point where we are comfortably paying for the website, and we're paying for the audio editing software that I use, and maybe buying Matt a beer every other episode or so. We've got lots of other things we could be doing. We could be spending more time on artwork. We could be doing more recordings, doing more mini-sodes, which we haven't done in a long time. So please send us your suggestions. You can also find us on Facebook and like us on Facebook, facebook.com/audiomunity. And, the main thing that you can do to support us, honestly, the best thing that you could do is to share us with your friends. If you have friends that are as nerdy about immunology as you are,
or you want to convince them to be.
Or you want to convince them to be. We've got a small and dedicated audience, but we could definitely grow a lot, and we need your help to do that. So if it's sharing directly, that's the best thing. But if you just want to go to iTunes or to Stitcher or wherever you listen to podcasts that has the ability to rate us, rate us, leave a review. It's super helpful to help other people discover the show. And, send us your paper suggestions through any of those contact links. You can find me on Twitter, blah, blah, blah. We've got links to all this stuff on the website. You're on Twitter?
Yeah, I'm on Twitter. I joined a long time ago, and I haven't tweeted yet, but I'm on Twitter now.
So
I have the app on my phone.
Follow other people, yes.
I follow a lot of other people. It's mostly politics, RuPaul's Drag Race, and The Bachelor, but I'm considering tweeting about science. My Twitter is exciting. Kate M. Franz, because that's my name. What's up?
Awesome.
Yeah.
We should put that on your bio on the website, immunity.org.
Like you could be Kate's first tweet.
Well, I have tweeted at one person. Just some thoughts about The Bachelor.
Our Patreons could get this as their...
but it's something I've been playing with, doing, joining this world of social media that seems to have taken everyone else by storm. Oh boy, why am I on a podcast? I belong in 1990. I found the Twitter. But I'm considering tweeting, so...
That's exciting.
So if you tweet at me, maybe I'll start tweeting back. Is that how it works? You tweet at people, right?
You can tweet at people.
Okay.
You can also retweet people. So if you tweet, maybe our listeners will hit the retweet button.
Yeah, I've also started liking tweets. Again.
Whoa.
Mostly politics, RuPaul's Drag Race, and The Bachelor.
Oh, is it not cool to like things? You should not like things?
No, that's totally acceptable.
Okay. I thought you made a no. Okay.
That's what I do.
That's how I the like once a week that I log into Twitter, that's how I know what to look at because of what other people have liked and retweeted that I'm following. Anyway, with that random digression into social media, I think we'll see you next time.
Sorry, I just never have anything to plug. I was really excited.
We're very excited for you.
Thank you.
Bye, everyone.
Bye.